So in this video, we're going to talk about the key ideas in flow cytometry as part of the F1 course on flow cytometry data analysis. So what we're going to think about is why you want to look at flow cytometry data with code. And our main reason to that is that we can deal with different data types. There could be fluorescence-based or mass-based data. There's lots and lots of tools and functions and packages and things that will help you analyze it. They've been written by people that are experts in that area, and allows you to use unsupervised methods that are non-manual and don't have any prior preconceptions of what to expect, and they're typically more effective than, say, two-dimensional gating. By writing code to do these analysis, whether it's very, very simple or very, very complex, you can use better tools and approaches and methods, more up-to-date methods. You can do reproducible work. So if you need to rerun things, that's fine. And that links to point 3, that you can tweak parameters and optimize things many, many times. You don't have to worry about doing things just once. You'll avoid laborious manual steps. That, sure, there's a little bit of learning at the start, but you actually become much more efficient and manage your time much, much better. And you also learn a highly transferable skill that's applicable in many diverse locations. So all those are good reasons why you should learn to analyze your data with code. So applying this to flow cytometry, there's certain key concepts that we're going to explore. The first is compensation. So this is the idea that there are certain fluorochromes that have overlapping spectra in terms of the fluorescence that's emitted, and therefore we need to subtract the estimated fluorescence from the other fluorochrome away from the one that's of interest. This links to using gates or gating. So gates are a series of cell types subset extractions based on the fluorescence intensity. So you might plot them against each other and identify cells that are of interest. And then we have these FMO controls, which are fluorescence -1 controls, and they're useful if there's multiple fluorochromes because empty channels can be used to measure the spillover of the fluorescence, and that can be subtracted then via compensation to get a more accurate estimate of the true intensity. So with this whole area of gating, visually we can think of it as you having an interest in these two cell populations here in green and red, and you might manually mark to know the area around which you want to select for further analysis and be able to look at those in a further graph where you look at them in more detail. And you look at those cell types only, which allows you more resolution on those particular groups that might be of interest. So in this way, this is kind of a supervised manual approach of gating. So what we're using in general terms is a mixture of unsupervised methods that are statistically based and applied in R, and then also supervised intervention from yourselves, the experts, on identifying, say, you have markers that identify known cell types, whereas you might have other cells that have an unknown cell type. There's elements of exploration, especially with unsupervised methods, whereas supervised methods, you might be trying to validate an existing a data set or sample set that are of interest. And here for the unsupervised set, you'd expect an unknown outcome where you're not sure to what extent the different cell types are different, for example. Whereas for supervised methods, you would expect that if you're using these in a reliable fashion, that the outcome is more predictable. So these kind of tie in together. They may have some cell types that are well known and some that are unknown, and therefore using both methods usually gives the best results for that kind of typical context. So in terms of our general steps, we can look at this diagram. So we're thinking about the pre-processing and preparing our data, and then analysis and modeling of that data and interpretation. So in terms of the first step of pre-processing, we think of this idea behind compensation. So that's subtracting for adjacent fluorescence. If we have multiple detectors, and typically we'll have many detectors, and therefore we're going to just correct for that effect. You might transform our data in some different way, and that depends on the underlying shape of the distribution. But typically that would be some sort of bi-exponential transformation to represent the data across two different detectors in a fashion that is focuses on the key components of variation within that data set. So obviously the details of how to do that would depend on the experiment and the data, so it varies a lot. There's also noise removal. So compensation is an example of that process. And then there's quality control as well, that we want to actually examine the data to see if there's some sort of deviation from that from certain cell types in certain experiments would include a control sample within experiments to allow better batch error detection across different samples. Then we can move on to the main analysis. So we could have dimension reduction methods that are typically based on around correlation. And we'll talk about those further. We can talk about maybe the levels of scales or the level of molecules that are something that's of interest. It might also be of interest to discover particular cell lineages. And there might be other features that are worth examining depending on what the goal is. And that will allow more quantitative associations between different features to be made depending on the idea. This is PCOA, stands for Principal Coordinates Analysis. And then there's also this idea that we have to manage our data more effectively. That means uploading it to repositories, backing it up when we need to for our own work, and making sure that's accessible for other people in our own lab, but also the community more widely. So these general steps link to the hurdles that we might expect associated with each step, and each step has some things to think about. So to think about at the start, the experimental design, the panel design, the settings, the instrumentation, and everything. Issues around spreading error, which we'll talk about further. The relevant control samples inclusion, and the details of the experiment to allow sufficient time, optimal time intervals for the right sort of fluorescence to be detected. And then we have the data preprocessing, including compensation transformation, quality control, removal of irrelevant events. So scaling the data also is experiment dependent, and that's something that needs optimization within each experiment. Following that, then we have potentially looking for different cell types or whatever the goal you have is, and that can be linked to cell markers if they're known, or known cell populations that are included in your data set. And then we have downstream analysis of whatever application. So we're looking at particular clusters of samples, particular features of certain dimensions, testing for changes in levels of different features as well. So that kind of completes this video on the key ideas in flow cytometry. Please see the next video on spreading error.